Assalamualaikum warahmatullahi wabarakatuh. We are from group 6 and we're going to present our presentation about observation plan of leaf spots in chili plants, analysis and explanation the sampling technique of leaf spot disease. So first of all, I will introduce you to our group member. So the first one is Safira, Tasya, Andrianisa with student numbers 171, which is myself, and Akhmad, Rudy, Fikri, Setyawan with student number 172, Sandi Setyawan Kertagosa with student number 173, Coryn Adelina with student number 186, Luhung Dharma B with student number 188, and Teksenia Saswati with student number 189. So the next one is description of plant. So the host of this disease is chili plants, and the disease itself named leaf spots. The pathogen that cause leaf spots is Cercospora species, and it caused by luxuriant less than 28 degrees Celsius temperature and 92% relative humidity, and the pH is 5 until 6, and below 90% relative humidity, the disease doesn't develop.
Disease description. This disease caused by the fungi Cercospora sp. It causes necrotic spot on the leaf surface, so that if the spot spread, it will cause the leaf to dry up to wither. So what's the aim? The purpose of the observation is to determine the intensity of the disease.
Leaf spots. Leaf spots are a disease caused by fungi named Cercospora sp. It is resulting in small necrotic spots on the leaf surface that widen, then causing the leaves to dry out, then fall off from the branch. Generally, occurs in the generative phase. It is affected by three main factors. There are susceptible host plants, virulent pathogens, and suitable weather. Next one. A scoring method was delivered by Mount and Lacey in 1982. It is one method of scoring criteria to determine the intensity of attacks based on the level of spread. 0 means asymptomatic or no symptoms detected. 1 defined 0 until 10% of disease spread on a leaf. 2 is 0 until 30% of disease spread. 3 is 0 until 50% of disease spread. And the last one, 4, defined more than 50% of disease spread. In this plot, we use 20% samples over the total population.
Number of sample. The number of sample plants is 20% of the existing population. So in this observation, we have 10 sample plants from 50 plants. Sampling patterns. We use zigzag sampling patterns because the fungi spread through wind, water, and vectors, so that the distribution is random, not clustered. Black point is non-sampling plants, and red point is sampling plants. Plants monitoring to determine the intensity of pathogen attack at a certain time. The location in Kebun Cabe Jogja, Jalan Wates Kilometer 9, Jati Sawit, Gamping, Sleman. Time. We start at 4th December 2020 at rainy season. And interval is 1 week and finish at 4th January.
The following is the data from the calculation of disease intensity, one block of 10 samples for 5 weeks. The average result of the disease intensity was 7.50%, while the average result calculated by the AUDPC was 6.56%. Following are the results of the graph of the intensity of leaf spot disease attack on chili plants caused by the fungus Cercospora sp. Result data is made in the form of a line graph showing the increase.
And this is a UDPC graphic showing a percentage increase over time. And the last one is our decision. So the composition and dominance of the pathotype in irrigation is only 7.50% from the result of the mean disease intensity, which means it is included in minor attacks, and chemical control is not necessary because the economic threshold is 40%. Okay, that's all from us. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.